Okay. Before we will start discussing the second topic, I would like to pass a little bit by some things I skipped last week. And I guess you succeeded to discuss it maybe a little bit, maybe more in your classes. Because the first thing we are starting with when we are coming into the microbiology lab is actually the lab itself, right? So which kinds of bacteriological labs do we have? Okay. First of all, let's be more specific. In microbiology, we can have different types of labs depending on different factors, very different factors. This is why, please pay attention. It's important in the entire life. It's important for tests, for exams, and so on. Pay higher attention to the fact how the question is formulated, because sometimes the way the question is formulated will help you to answer the question. In the most of cases, the answer is in the question. Sometimes the question is not specific enough. In this case, you have all the right to give extra questions. Because when I ask you, please give me classification of microbiological labs. Usually you start answering with classifications that are more familiar to you, right? But in this case, it would be fair to ask to classify them or to arrange them in order to what? Because microbiological labs can be divided in following groups according to their target, or according to microorganisms we are working with, or according to the reason why we are making investigation. For example, if we are talking about the pathogen, microbiological lab can be bacteriological, virological, mycological. These three main fields. If we are talking about the target of this lab, or let's be more specific, for which reason we make those investigations, it can be scientific lab, it can be study lab, as in a department, it can be a clinical lab, and all of them are having different targets. Here, we are using this lab in order to learn microbiology, general microbiology. In hospitals or nearby hospitals, we have clinical labs. Clinical labs have the only target to find out the pathogen, if we're talking about microbiological labs, because also we can have separately clinical labs, and please don't misuse them, because sometimes when we are talking about specimens, for example, we take from patient for investigation, and you say, we take blood for investigation. Okay, and what you are doing with that blood? General test, routine test. Okay, it's for a clinical lab, not for a microbiological lab. You can make general investigation or routine test of the blood. You can make biochemical investigation of the blood. You find out other parameters. You can make bacteriological investigation of the blood if you suspect presence of some bacteria in the blood. Absolutely different type of investigation. We can make serological investigation of the blood. In this case, we are looking for antibodies in that blood. Absolutely different type of investigation. So, sometimes we are talking about scientific labs. Scientific labs are the mostly amazing labs from point of view of variety and opportunities, and I would say in most of cases financial limits or their lack of those limits. And at the same time, apparently mostly useless labs. Why? For example, some time ago I have been in a conference on which there was a representative of a scientific lab from Europe, and the professor and the director of that lab was talking about rare pathogens, rare infections they've been studying in that lab. And since most of our colleagues are coming from hospitals or from the university, some of them asked, what's the reason to waste that much money to study infections you even don't have in your country? And he told, this is the mostly amazing thing about scientific lab. You can do there whatever you want. Of course, in legal limits. But you can do there whatever you want. It is not about efficiency, especially financial efficiency. It is about finding more knowledge about different pathogens. Now, how can you use those knowledges? It is another issue, another matter of discussion. But still, scientific labs, they have absolutely different targets in comparison with clinical labs. And now, in clinical labs, you will have pretty specific types of investigations. You can be amazed how many kinds of ways of investigations we can make. Just as I told you, general, biochemical, bacteriological, serological, and so on. And all of them are also divided in a lot of kinds of investigations. You can barely keep them in mind. And at the same time, in one lab, in one clinical lab, you can visit even here on the Malina Mica. You can find out there is the same list of investigations you can make, absolutely, most of them, no difference between them, and they do nothing else. They have strictly their limited list of investigations, and if you are asking for something else, no, sorry, it's impossible. Because we are working only with those ones. Or they will say, we can do it, but it would be much more expensive. Why? Let's make comparison with pandemic situation. Do you remember how much was to make a PCR test in the beginning for COVID? Did you get that period? You've been here or home? You've been home. Did you get to the situation you need to make a PCR test in that period?
Yes.
Some of you yes, some of you no. Do you remember how much it was? Huh? Extremely expensive in comparison with most of investigations you are doing on a daily basis, it was extremely expensive. For example, here in Moldova, in the beginning of pandemics, first of all, we've been so happy when first 100 kits for PCR test came here in Moldova. Because you can have PCR machine in your country or in your lab, you have it, but if you don't have specific kits for this very pathogen, you cannot make any investigation specifically for this infection. So when the first 100 kits for PCR test came in our country, everyone was like, finally. And what is 100 tests even for such small country as Moldova? It's nothing. For example, only in the hospital I'm working in, about 600 people are working. And it is not the biggest hospital. So you cannot test even a quarter of staff in the middle hospital, in the capital. But still, everyone was really happy about that. After that, but there wasn't any discussion about like commercial testing for everyone. It was simply impossible. First few tests, we've been collecting samples, can you imagine? Sending via DHL or FedEx to, I don't remember, either Romania, either Germany, I don't remember, to make those investigations, and in about 2 or 3 days we've got results. First few cases we discovered in this way. Because it was the mostly wanted product on the world market in that moment. And when they started producing that, it used to be extremely expensive. So when it became available for general population, one PCR test was about 50 euro. Something like that, one PCR test. If we're talking about antigenic test, if we're talking about antigenic test, in this case, it was about maybe 20 euros or 15 euros. Okay, much better, but still, in a lot of places, it was required specifically PCR test because nobody believes nothing but PCR test, especially in the beginning, right? Like, it is science, and this one, like you went to astrologist and got a paper from non-authorized person. Now, we almost don't use anymore PCR test. We only use rapid test for antigen or for antibodies. It depends what we are looking for. So, later they came much cheaper. Right now you can go in a regular pharmacy to buy rapid test for COVID. It's gonna be about 20 or 30 lei, one test. And in the beginning it was 300 lei. Okay. That's the difference. Why it is so?
Because the bacteria isn't as rapid as it was before. Why the test is no longer expensive as before is because it's no longer like a deadly cost to the world.
On the first place, we have market. On the first place. And I guess you agree with me that there is almost no one in this world who didn't try to make profit on this situation. right? From the very regular person from those up to us. Yes. And in legal limits, that's absolutely normal. That's absolutely normal to try to make profit from everything you can in this world, in legal and in ethical limits. But beyond financial interests, imagine, in the beginning, this test wasn't that required. So few samples or few examples, few kids could be present, like few thousands for the entire world. And you need to make huge production. You need to produce a lot. As less you produce, as more expensive is production. As more you produce, as cheaper is production per piece. This is why if you want to buy something in huge amount, you pay less, right? If you want to take only one piece, it costs more. Or masks. Do you remember how hard it was to buy a mask in the beginning? Pharmacies were out of the mask. Me personally, I ordered on AliExpress a box of 100 masks, and till now I didn't receive that. Yes, because afterwards I found out from news that US stopped a bunch of ships going from China to Europe and to other countries with cargoes, with all of these equipments, including masks. They stopped and brought them in US. I'm still wondering, did they pay it or not? But everybody, by any legal or illegal way, were doing such things, looking for their profit. So it was almost impossible to buy them. After that, I'm pretty sure at least some of you still have some storages of masks home, right? Me too. And what would you like to sell them? Same thing with tests. They sell them out because they don't need them anymore. With that small price. Actually, with the price they produce them. Maybe a little bit more, but not too much. And one more thing. For example, which is the mostly common car for India? The mostly common, affordable car in India? Tata. Okay. Which is not really a common car, but still affordable. Like, it is cheap, but it is not really common for India. Huh? Suzuki Swift, right? Okay. Or other Suzukis. It is not that common. Okay. Meanwhile, they're quite not too much expensive. Both, right? Either some of them could cost the same, right? For which one parts will be cheaper? For which one parts gonna be cheaper? Who is for Tata? Who is for Suzuki? For which one parts gonna be cheaper? Why?
Because it's manufactured in India itself.
Huh?
It's manufactured in India itself.
And Tata?
Tata too, but Tata is a bit expensive than Suzuki.
Okay, that doesn't make too much sense. you have ruined the entire speech. Because, okay, I will move to our situation. Too much Toyota's Prius we have here. Toyota Prius is very common here. Still, with the same price, you can find some, for example, Opel. Also not too much expensive car, even cheaper than Toyota. Toyota is a quality. Opel is okay. So, in most of cases, parts for Toyota Prius gonna be cheaper. Why?
Because it's more
It's very common, and we have a lot of places, including also not manufacturing. We don't have manufacturing, but we have a lot of disassembled cars, and you can buy cheap parts, good parts, original parts from disassembled cars. So that's the reason. As more common is a product, as cheaper it's gonna be. Both the product itself, because if a lot of people wanna sell the same product, there is competition in decreasing of price. And including maintenance. It's gonna be much cheaper. You can afford it easier. Same thing with tests. If we need a lot, their production, their manufacturing, gonna be much cheaper. The cost of one piece gonna be much cheaper. But if you don't really need tests for Ebola here in Moldova, first of all, I'm almost sure it's gonna be almost impossible to find a test for this infection. But if you find it, it's gonna be 3, 5, 7 times more expensive than tests for COVID or for flu or for any other infection that is much more common here in Moldova. This is the reason, the simple explanation why prices are going up and down. And in other countries where flu isn't that common and Ebola is much more common, there is a chance the tests for flu are going to be as expensive as here for malaria or something like that. And there, it's going to be much cheaper, or even for free. Yeah.
in our places for free. Because Ebola was kind of common with malaria too.
Yeah.
So it's very cheap.
Yeah.
Or free actually.
So it makes much sense. Okay. Now, if we're talking about that classification, you already described BSL 1, 2, 3, 4. I guess you already know it, right? How we decide how we classify different labs?
We have biosafety labs.
Yes.
We have the I don't know how to pronounce other that's not for biosafety, like clinical labs, industrial research labs.
Okay, we have 4 levels. 4 levels. BSL 1, 2, 3, 4. Biosafety level 1 is the simplest one. Teaching labs we use for studying, including our lab here in the department. So BSL lab number 1, it doesn't require too much precautions. You can see you are not required to wear mask or respirator or gloves or cap or anything else. You're required to wear only the white coat, and even this one is not that important from biological safetiness perspective as from teaching perspective. You should be used with white coat. If you are in a medical university, instantly it shows your attitude and your responsibility. Faster, please. Your responsibility when you are coming in the class on time, with a white coat, participating, answering, asking, and so on. Right? But there are no much risks here in BSL 1 because mostly we are working with non-pathogenic or conditionally pathogenic flora. What does it mean? We have generally 3 kinds of bacteria from this perspective. Non-pathogenic, or also we call it saprophytic bacteria. They are participating in decaying of organic matter all over the world. They are not bothering us. We are not bothering too much them. We produce only benefits to each other. They are participating in circulation of organic substance in the nature. They are participating in our digestion, and they help us to digest some complicated substances and even producing some vitamins and so on. So we produce only benefits to each other. Even after our death, we produce food for them because they are participating on decaying of dead bodies of humans, animals, plants, and so on. We have strictly pathogenic bacteria that are responsible on our diseases. There is a big difference between them because strictly pathogenic bacteria are extremely susceptible to the environmental factors. Extremely susceptible, with few exceptions. There are some strictly pathogenic bacteria that can easily survive in the environment, but most of them are really susceptible to the environmental factors. That means if the temperature in the environment will increase much higher than they can support, or decrease much higher than they can support, they're gonna die. Or at least they will stop multiplying. And little by little they start dying. But when they get into the host, in the conditions they feel mostly comfortable, they start multiplying as fast as it's possible and producing diseases. And we have conditionally pathogenic flora. They are situated somewhere between non-pathogenic and strictly pathogenic bacteria. Usually they are making function of normal flora, but for some reasons, in some special conditions, this is why they're called conditionally pathogenic flora or opportunistic flora. By some opportunities, they are able to produce some infections, some diseases. So in BSL 1, we are working only with non-pathogenic or conditionally pathogenic bacteria. Both of them are normally present mostly everywhere. Around us, on surfaces, on our skin, in our hair, in our mouth, in our digestive tract, almost everywhere. BSL 2, it's a regular clinical lab. There we are working with the mostly common, but not highly, contagious infections. For example, some basic skin infections, like infections with Staphylococcus or Streptococcus, some food poisoning, general food poisoning. Not all of them. How you understand which one is more contagious?
When they are transmitted by air.
Okay, makes sense. Why Why infections that are transmitted by air are more contagious than others? Because we breathe, exactly. Are you making any breaks in breathing? No. 24 hours, 7 days a week, we are breathing without getting tired of that. Did you ever think about it? You never get tired breathing. And it makes us a very good source of infection if we're infected. And at the same time, the same fact we're breathing all the time makes us extremely vulnerable as a receptive organism. On the second place, we have infections with transmission by food and water. And the principle is the same. For how many times you eat and drink? Not as much as breathing, fortunately, but at least every day. And probably not once a day, right? Now it depends on anyone's habits, but mostly every day and mostly more than once. Are you washing your hands before every meal? Are you confused because of the fact you recognize it, or because most of them are not able to recognize the truth? I guess there is no person in this world who could say, I wash my hands before every meal. If you're going to be truthful, right? Because now it depends. Now it depends what meals, what meal means for you. Because if you call the meal only the moment you come home, you put a plate and you start eating soup, okay, that's meal. Okay, probably in that case you wash your hands before meal. But do you call the meal the moment you go to the break in the coffee shop to buy a piece of placenta or a cup of coffee? Do you call it meal? Is it a meal? For how many times you have such a meal? At least once a day, right? How many times you wash your hands before such a meal?
Never.
Mostly never. Mostly never. And it is a real student's life. It's a real student's life. Did you ever think about this? Probably not really. Fortunately, we had pandemic. In pandemic, mostly from any place you heard, wash your hands, disinfect your hands, wash your hands. It's extremely important in order to stop spreading COVID, even if it is not so. Did you know it? Did you know that washing hands doesn't help too much of stopping spreading COVID? And what about those scientists who've been talking like that? Are they aware about that? Already yes. Already yes. But what we realized after pandemic, not too much people knew you have to wash your hands on a daily basis. They found it out finally. Yes, because during pandemic dramatically decreased incidence of intestinal infections due to this fact. It didn't decrease too much COVID spreading, such as vaccination, such of isolation specifically when you're sick, such as wearing the mask especially in the environment where you can potentially meet a sick person. Those measures are much more efficient than washing your hands. Of course, hands hygiene decreases somehow, but you will never absorb it so much. But talking about intestinal infections, it is extremely important. Personal hygiene, especially hands, because you are doing mostly everything with your hands. Everything. The mostly clean things in your life you are doing with your hands. The mostly dirty things in your life you are doing with your hands. Did you ever realize that? This is the reason why on the second place we have intestinal infections, or infections that spreads with water or food. Next place is... Huh? No, on the first place. They're on the first place, airborne infections.
Environment?
No. Contact, exactly. We are social. We contact to each other. Skin infections, sexually transmitted infections, infections that are caused by direct or indirect contact. Yes, starting with the mostly general, like, did you ever have pimples? It is also infectious process. It is localized, extremely small, but it is still infectious process. Starting with this one, who had everyone, and finishing with such as HIV, as hepatitis, B, C, D, tuberculosis is airborne infection. So, on the next place, even if you're sociopathic, extremely sociopathic, and you're trying to distancing yourself from the entire world, it's impossible to do it 100%. This is why next as event you will have, you're breathing much more, you're eating less and drinking, and you are contacting to each other even less, but still a lot. And after direct contact, we have indirect contact. What means indirect contact? Somebody before me have been touching this table. Somebody before me have been touching that bill. Somebody before me have been touching this door. And so on, and so on. And you never know who've been touching and what did that person do before. And a special public transportation. How do you feel when you... It's more safe. It is harder to remove your hand from there and you will never fall. But still. If it is not really uncomfortable for you touching those handles, ask yourself, what about your personal hygiene? Maybe because you're used with that. Maybe. But overall, most of people can feel difference between their home surfaces and outside, right? And on the last place, the last place, infections with parenteral transmission. What means parenteral transmission? Parenteral transmission. Yes. Parenteral, not parental. Parenteral. The origin... you know what is Paralympic Games?
Yeah.
Paralympic Games, you don't know it? Olympic Games, you know, I guess. Paralympic Games are games that are happening after official Olympic Games, but for people with disabilities. And this root, this is a complex word made of two roots. Para, that means beyond or nearby, and central word is the next one, Olympic Games. Same thing, para-enteral. Enteron in Greek means intestine. Okay. Para means beyond or bypass or nearby. What does it mean? Mostly everything normally gets inside of our body through the intestinal tract. Mostly everything through the intestinal tract. But there are some things we can get either by purpose, either by an accident, in the parenteral way. It is everything regarding through the blood. Injections, surgeons, some traumatic events, and so on. So you can get something inside of your body, either through the classical way you're doing it every day, either through the other way. It is not that common for you. Who had ever surgery? Not everyone. So even in your life, now I guess most of you are about 20, right? During 20 years, it's a big period of time. During 20 years, most of you didn't have any surgery. How safe you are from this perspective. Can you imagine? Okay. Let's go next. Who didn't ever visit a dentist? 2 students? 3? Not bad, but already much less. You see it? Same. Okay. Who has piercing? Bless you. Who has piercing? Yes, but you did it. You did it. Yes. Okay. I'm not blaming anyone who is wearing it or who is not. The question is, you passed through this moment. You passed through this procedure of making piercing, right? Who has tattoos? Tattoo. Not too much. Makes sense. Usually, what I observed, people with dark skin, it doesn't make sense for them to making tattoos, right? Not too much. Huh?
I'm not black, so I could.
No. I say I.
I'm joking. I'm sorry. I'm joking.
Okay.
I'm sorry.
Don't make me that stressed out. So if I'm wrong, please correct me, because I'm not. Yes.
It's kind of dirty when you do it.
It doesn't make too much sense for people with dark skin to make tattoos. First of all, it wouldn't be that visible. And usually you are doing it for show off, right? Everybody should see how cool you are. And usually you are doing it in a period of time you are pretty damn, like teenage or a little bit more. Okay. This is category of people who are not really used with that. And one more category, religious people. Religious people are not really used making tattoos, right? And I guess somehow here we have either from that, either from another category, either from both categories. This is why we don't have too much people with tattoos here. But still, I've been surprised to discover for myself that you are going you're walking in the city, and it is almost impossible to see a girl without a tattoo. It's much easier to find a guy without a tattoo, but not a girl. I've been really surprised about that. Here in Chisinau. Yeah. So it is one more way. What is in common between surgeries, piercing, tattoos, dentistry, invasive procedures that are deregulating integrity of our soft tissues? Any such event is a potential is potentially opening a portal for entry of the pathogens, bacteria, viruses, and so on. So even if you are extremely addicted to piercing, tattoos, you are going often enough to a dentist, still those events are not be as often as breathing, eating, drinking, and contacting to each other. This is why this one is on the last place. And you will observe that in nature we have a kind of balance. As higher is contagiosity of infection, as lighter is infection. As higher is the chance to acquire this infection, as usually infection is lighter, you can easily pass through it. As much more rare is infection, as more complicated the way of transmission, as more aggressive is this infection, and as higher is mortality for such infections. So there are some infections you cannot control. You cannot really control. And usually it's about airborne infections. That means there is almost no chance to not acquire those infections. In other words, we can say, in most of cases, it is not your fault. In most of cases, it's not your fault you acquired that infection. This is why nature gives you a second chance, or much more chances than only a second one. But as more responsible you are on your behavior, going down, personal hygiene, personal habits, contacts, and some different invasive procedures, including, by the way, not only everything we described, but also using of drugs. Same thing. And here is the highest rate of spreading of such infections because of a lot of factors, too much factors to describe all of them right now. So, as less chances you are giving to yourself, as less chances nature will give you, or whatever you believe, universe, God, whatever, right? So all of us are really responsible on consequences we are going to. Okay. So in clinical labs, BSL-2, we have, what do we have? Something that we can easily contract, something we can really easily acquire. It is not something extremely contagious. It is something really common. It is not something extremely aggressive as disease. Mostly common infections. BSL-3 are extremely contagious, but not extremely aggressive. For example, all the airborne infections, viral, bacterial, starting with common cold, flu, COVID, and finishing with mostly aggressive one, tuberculosis. We have nearby diagnostic center, we have a special lab for tuberculosis. And for BSL-2, for example, you're required wearing gloves, you're required wearing mask, you're required washing and disinfecting your hands every time before and after all the procedures you are making, all the manipulations you make in that lab, and nothing much more. Some special type of ventilation in that lab, but nothing extremely complicated. Basically, almost anyone, if we are talking about bacteriological lab in a hospital, BSL-2, mostly any staff can enter and leave that lab easily without extra precautions measures. When we are talking about BSL-3 and BSL-4, you need first of all, we have a bunch of restrictions, requirements, equipments, and procedures we are doing before entering and after leaving the lab. If you will have ever opportunity to visit BSL 3 or 4, you will observe that they have separate locker room. They're coming from outside, they're changing their clothes, they're washing their hands, they're changing their shoes. They're going to a special cabin. Nearby that cabin, they unpack new sterile suits, like in COVID, you saw it. They put on those suits, they enter that cabin, one door, they disinfect their hands one more time, they open another door, they close it, they're in the red zone. And in that place, with respirators, with goggles, with everything, with shields, with suits and few pairs of gloves, they are going in the red zone to work with pathogens. After they finished working with pathogens, also, yes, in BSL 3 and 4 we have a special artificial ventilation with incoming air and outgoing air, like pumping air through filters, through special disinfecting filters. So mostly sterile air, basically almost sterile air, is going inside the lab in order to decrease the chance of contamination of those substrates they are working with, and to not let pathogens leaving this lab through ventilation also. They're coming and going out through filters. Of course, we will have UV lamps all over the lab, and especially in the zone we are working in order to make, from time to time, to make that disinfection with UV lamps. And after that, after the person is finishing working in the lab, staff is going back to that door. They enter between doors. There is a place they can throw out the suit. I guess you saw how it's easy, how it's correct, or you didn't, how it's correct to remove all of those suits. On every step, they are disinfect their hands. First of all, you disinfect your hands, you take off your gloves, you throw them out. You disinfect your hands. You start opening the suit, taking out the hood, taking out goggles, throwing them out. Again, disinfect your hands. You open it and you roll it from outside to inside, you know? Like to make them to make contaminated part inside. And after that, close it, and close it, and you disinfect your hands again. This is what they are doing with this suit. What happens next? They are leaving. They are disinfecting again their hands and opening the second door and leaving that box. This is how it works in BSL 1 or in BSL 3 and 4. Now the question, if there is no much difference between how we are equipped in BSL 3 and 4 and how is organized BSL 3 and 4, why do we have difference between them?
BSL 4 is for eradicated and highly dangerous diseases, polio and Zika virus.
Okay, so very good. The equipment will be pretty same. The requirements for the lab, for the building, and for space will be pretty same. But difference will be the pathogen. Because it is extremely important to keep them inside, to not let them getting out. And since both one of them are extremely contagious, but not that dangerous, like flu, COVID, tuberculosis, and some of them are less contagious, but still extremely dangerous, like Ebola, Zika, and so on, because mortality in such infections is higher than 90%. Higher than 90% is mortality with those infections. This is regarding types of bacteriological labs.